We're going to look at osmoregulation in plant tissue, and we're going to do an experiment with vegetables and indeed fruit to investigate osmosis in different concentrations of salt solution. Pause to look at definitions of electrolyte, osmosis, and osmotic pressure, and the words hypertonic, isotonic, and hypotonic. In hypertonic, water leaves the cell, so the concentration outside the cell here is more concentrated, so water leaves the cell and damages the cell, which is plasmolysis. The isotonic, where there's the same amount of water going in and going out, because the osmosis, the movement of water, is going to take place, but the pressure stays the same and in an ideal, what's called the flaccid cell. Hypertonic, greater water ingress, and that's because the cell has greater concentration of solute than outside, so the water will go into the cell, gets larger. In your lab, you're going to choose your vegetable and you'll be supplied with a 1 molar sodium chloride solution, which you will dilute into 5 different concentrations. You will calculate the percentage mass change of your vegetable sample in the different salt solutions. And you can pause to predict the following as given there. You're going to choose a cutting material. All of your apparatus and equipment, you're going to give a list at the end of the second lesson as to what you want for your lab. You will also have the reaction vessel, the containers that you're going to place your sample in the salt solution. And pause to look at the other materials that you may want and anything else that you need. And so first 2 lessons, planning and research, read that very carefully, and those to be completed by the end of the second lesson, and to give in your material equipment list. So it'll be ready for you for the 2 lab classes, and including some of the write-up there. And then 4 days later, hand in your write-up and analysis. So pause to read each of these, what you're actually doing with your 5 samples, describing what they look like, how you make your solution, and that you're going to mass and record each sample, how you're going to give letters to them, and leave for at least 3 hours, noting the time, and you'll dab any extra liquid off them before you mass them at the end of the time period. Calculate the percentage mass change and graph your results, but pause to predict
That's it.
Guess you have to cut 15 pieces. You have to cut 15.
Yeah. Okay.
It doesn't matter, the size doesn't matter, right? No, I don't know. We want the water is like these. Yeah. Here's over. Yeah. Oh, just pure distilled water.
1.39 goes into 0.4 moles 2.
It's fine, we're gonna cover that.
And 1.43 goes into 0.4 moles 3.
And then half of the water measures 0.1, right?
1.33 goes into 0.4 moles 4.
What is it like? 1. But you said he's sick, right?
What you think it'll look like before you actually plot it. And here we're looking at the solvent, because it's the solvent that moves through that selectively permeable membrane here, the dotted line of the cell wall. And where there is more water, it's more dilute solution, and it will pass to where there are fewer water molecules, where it's more concentrated in the diagrams here. And pause to answer the question in red at the bottom there. So again, some more definitions to pause and to read what's happening with osmoregulation and osmotic pressure. And another diagram showing the movement of the water. The left-hand one, hypertonic, going from where more water is actually present, so a more dilute solution, into or out of the cell, where there is less water, more concentrated solution. Isotonic, water is moving in both directions at the same rate. And hypertonic, water solution is surrounding the cell, is less concentrated, and will pass through the cell wall where the solution is more concentrated, where in the diagram here less water is shown. And pause this one for a while, because here put the sodium chloride ions in as well. They do not go through the cell wall, the selectively permeable membrane. It's only the water that goes through from a more dilute solution to a more concentrated solution area. And here you can pause to see more detail on the cells and an actual photograph of the microscope of an aquatic plant, Elodea, in hypertonic solution, where the center of the cell has shrunk because it's lost water to its surroundings. And pause for each of these to find out more. The second one, incredibly expanding vegetables. Then effect of salty soil on plants, how it affects the plant itself. And for animals, osmosis in fresh water and salt water. And here it's the movement of water and ions, but the ions won't be going through a selectively permeable membrane. And you can look at that and remember that with the saltwater fish, you've got salt solution around it and what the differences are there. And here in human blood cells, red blood cells, and how hypertonic, they're shriveled. The water has traveled through the cell wall greater amounts, and shriveled isotonic, where it's just right, and hypotonic, where the cells are swollen because more water has gone into the cell. And here in paramecium, the contractile vacuole here, because you'll get osmosis through the actual cell wall, outer wall of the paramecium, and you can see the animation of the contractile vacuole and follow that. Pause to look at more experiments, naked eggs in water, and parts of plants that move because of osmosis. And many thanks to Miss Hume and her IB biology class and Washington International School Science Department. Thank you.